Welcome. Welcome at the Associate Professor Lecture organized by the Strategic Program Cancer of the UMC Utrecht. Associate Professor Lectures are being organized to celebrate the appointment of a senior researcher as Associate Professor at the UMC Utrecht. These lectures are intended for a bigger audience, not only the UMC scientific colleagues, but also patients and of course family and friends. I am Nico van den Berg. I'm a professor of computational imaging in the Division of Oncology and Imaging, and I'm also a member of the Executive Board of the Strategic Program Cancer. Today I have the honor of introducing Dr. Ingenieur Trudy Straetemans on behalf of the program. Trudy graduated from Wageningen University with a Master of Science in Animal Sciences with a focus on immunology. And she obtained a PhD at the Erasmus Medical Center in Rotterdam. There she was trained as a tumor immunologist and focused her research on T-cell gene engineering strategies against cancer. Her long-term research is driven by the intriguing interplay between cancer and the immune system, and she dedicates her career to bringing immune intervention strategies against cancer into clinic. In 2012, she started as a postdoctoral fellow at the laboratory of Professor Jürgen Kubal. Currently, she's Associate Professor and PI at the Center of Translational Immunology, leading a gene and cell development team for ATMP development. And ATMP stands for Advanced Therapy Medicinal Products. Trudy has been exploring means to decrease the development time of advanced therapy drug developments at the Utrecht Science Park, which has resulted in the contribution to the design of ICAT, Innovation Center for Advanced Therapies, which is implemented in a national growth grant, pilotfabriek voor regeneratieve geneeskunde van RegMed XB, granted by the Ministry of Economic Affairs and Climate. She believes that the collaboration across disciplines is key to bring new technologies in medicine forward, and therefore she was one of the initiators of the DARE-NL infrastructure. DARE-NL is a KWF-funded Dutch infrastructure consortium between 11 partners in academic ATMP development and just celebrated its first year anniversary with a successful symposium. So it's very clear that Trudy is a very qualified and engaged researcher, and we are very pleased and very honored to have you here at the UMC Utrecht. Trudy, your lecture is entitled A New Era, Implementation of Living Drugs in Clinical Practice. The floor is yours.
Thank you, Nico, for this kind introduction, and thank you to the Strategic Program of Cancer to give me the opportunity to discuss with you my work today. First, I would like to introduce you two cycles that form the basic of my research. I will start with the cancer immunity cycle, where you see in a simplified way the interplay between cancer and the immune system. Below are cancer cells dying, releasing their antigens and present it to the immune system. Antigen-presenting cells in lymph nodes are activating T cells that then traffic towards a tumor site, migrate there and infiltrate in the tumor tissue where they can recognize and kill tumors. Often this process is taking place unnoticed and there's nothing to see. In case this process is not going well, the patient presents with cancer. How nice is it to use these elements of the immune system to harness our own immune system in the fight against cancer? Examples of this are so-called checkpoint inhibitors, antibodies that are very effective in a subset of patients. Or another strategy is T cells that are engineered to express an immune receptor, such as CAR T cells, a nice example of living drugs. But in order to get these medicines to our patients, we have to go through another cycle, the drug development cycle. When I joined the UMC Utrecht, together with a great team, we went from discovery and development through preclinical research into a clinical development and a clinical study. In my associate professorship, I would like to combine the expertise of these two cycles and bring new medicines to our cancer patients. And with my expertise in drug development, I would also accelerate translation of new living drugs beyond the field of cancer. So going back to the cancer example and the living drugs consist of fully functional cells that have been selected and are often modified to treat specific diseases. An example are tumor-infiltrating lymphocytes, isolated from the patient, expanded ex vivo and then reinfused into the patients, or cell vaccines made from specifically conditioned immune cells or cancer cells. And if we zoom in, the CAR T cell example, a schematic picture illustrates a CAR T cell cycle where a chimeric antigen receptor that recognizes the tumor is introduced in the T cells of the patient, expanded and then reinfused into the patient. So now we not only have synthetic drugs, biologicals such as antibodies and growth factors, but we also have living drugs in our medicinal toolbox. These are classified as ATMPs, Advanced Therapy Medicinal Products. And by the EMA, they are defined as medicines for human use that are based on genes, tissues or cells and offer groundbreaking new opportunities for the treatment of disease and injury. If we dive a bit deeper into the classification of ATMPs, we see in the middle the cell-based medicinal products that I just described. And if they are genetically modified to express an immune receptor, for instance, they are classified as gene therapy medicines. This classification implies that these need to be manufactured in compliance with good manufacturing practices, so-called GMP. And these GMP guidelines for ATMPs are a specific chapter in the EU GMP guidelines. These medicines, this also implies that GMP facilities in hospital centers are crucial for the translation of these drugs. So how do we now prepare for a first-in-human clinical study? I will show that by our TAG example, where we got approval for in-house production of a gene therapy medicinal product. Shortly, the concept of tags. Tags are T cells engineered to express a defined gamma delta T cell receptor. So gamma delta T cells are a particular subset of T cells present in each individual. We found in the lab that individual T cell clones were able to recognize a broad panel of tumor cells. And from those highly potent tumor cells, we isolated the genes that encode their receptor. We transferred them in a retroviral vector, and in that way we could express them in normal T cells. Once they are expressed on the surface, we see that the endogenous receptor, the alpha-beta T cell receptor, is downregulated, and then we call them tags. So why using gamma-delta T cell receptors? That's because these receptors recognize a hallmark of cancer, which is deregulation of metabolic disorders. In contrast to normal conventional alpha-beta T cells that, dependent on the genetic background of the patient, recognize specific individual mutations that we call neoantigens. So when I joined the UMC Utrecht, the TAG concept was just published. We started to identify the lead receptor, and others were working on the mode of action. So I started to improve the production and purification method, and that led in an increased tumor efficacy. And we developed side by side a variety of models to test efficacy and safety. This all led to the first part of our clinic, not only to nice papers, as you can see, but also to the first part of our clinical study dossier, the investigational brochure. Then we continue to translate our production protocol to a GMP-compliant manufacturing protocol, and we looked at the molecular level to our transgene from beginning to the end of our product. That resulted in the second part of our clinical study dossier, the IMPD, that contains the quality manufacturing and control of a medicine, and a license to conduct a study in human with genetically modified organisms. This is the study setup. Simplified, we see that we include refractory AML or multiple myeloma patients. T cells are harvested. They are engineered to express in green the gamma delta T cell receptor. They are expanded and purified, and we reinfuse into the patient after 2 weeks a pure population of tags. So I just want to show you, take you along in how you translate a manufacturing process from the bench to a GMP compliant process, and that's what we did in the UMC Utrecht cell therapy facility. So we have our concept, the tags, and together with the scientists you define what are the critical quality attributes and that we translate in release criteria. We look into detail in all the raw materials and starting materials that are necessary for production, suppliers, and then we look into the assays that are necessary for controlling the quality of the product, and then we after that we validate in technical runs first, and then in product validation runs these assays and the process. This all leads to documentation in the IMPD, but also standard operating procedures, worksheets and labels are being finalized in order to get regulatory approval. Training and qualification of personnel is being done along this route, as well as documentation. If we zoom into our tech manufacturing scheme, we see that first we have our apheresis. The cells from the patients are cryopreserved and need to be released by a qualified person before we can enter the manufacturing process. At day 0, we thaw the cells and activate them. We can only continue when we have enough T cells. 2 days later, the viral vector with our transgene is introduced and cells are expanded until day 6, whereas an important in process control, we need to check whether there's enough transgene on the cells. And then at day 13, the end of the procedure, we harvest, concentrate, and release the product. This is an example of an in-process control. We want to have more than 20% of the cells that express at day 6 the transgene. And here you can see that in these graphs we are looking, you see that we have more than 50% of engineered cells. These data are from technical runs and validation runs. On 2 different days we evaluate the number of tags in our product. And we also look at expansion of the cells. At the end of the process, we have parameters that need to be addressed. So we have the identity, the purity, the impurities, and the sterility. And our cells, after purification, look as at the left right graph. So that led to regulatory approval and the start of the study that we run together with Monique de Witte, who is the clinical PI. It took us, until now, 15 years. I want to share some clinical results of our dose level 1 and 2. In dose level 2, we see clinical responses, a stable disease of more than of around 12 months, and even a complete remission that lasted for 6 months. We also could find back in our patients our tags. And all these promising results led to decide that we escalate to our next dose level 3, where we're currently recruiting patients. I believe that we need to work together with many disciplines to accelerate translation. And that's why we got the GMP simulator facility designed, which is part of ICAT, an innovation center for advanced therapies. It is there are 3 different facilities in ICAT. We have the GMP facility, our GMP simulator facility, and the biofabrication facility. Zooming in the GMP simulator facility, we offer the following services to speed up translation: training by experienced and qualified personnel, process development support, analytical method development and validation support, and regulatory support. These are examples in the simulation facility, such as a CAR T-cell manufacturing for pediatric patients and adult in a collaboration with the Prinses Maxima Centrum and the UMC Utrecht. We are setting up viral vector technology development and GMP production of viral vectors. Together with Bart Spee, we're tackling donor liver shortage in a new bioreactor in the GMP simulator facility. And we're also looking into new technologies which are non-viral gene editing for immune cells. But also at national level, we are joining forces together with the Radboud UMC Harry Dolstra, the UMCG Edwin Bremer, the NKI Inge Jedema. We initiated DRNL, and DRNL consists of 3 pillars: the ATMP knowledge framework, where you can see DRNL data, training and valorization, GMP production process harmonization, and QC harmonization and assay development. We have our ATMP Biologics and Technology Hub, where we have the viral vector platform and future ATMP technologies, supported by our regulatory, health economics, health technology assessment, and patient access pillar. And when we look into a broader picture, we would like to emphasize that DRNL and ICAT are part of a broader ecosystem where here the basis is here OnCode Accelerator, also a recently funded growth fund, where we have go from target, patients, artificial intelligence to organoid and models, clinical point of proof of concept trials and drug development. And ICAT and DRNL are fitting in this ecosystem, nicely connected with other infrastructures like NextGen, where we look into equipment and technology development, a stem cell infrastructure, and another cell and gene therapy infrastructure. And we connected together with another infrastructure fast. So I would like to thank the following people. First of all, Jürgen Kubel, who was the inventor of the tags, Scholt Sebastian, with whom I work together already for a very long time, Monique de Witte, who I'm running the clinical trial together, and Dennis Beringa together are completing our team of PIs that leading the research group that you can see on the picture. I want to highlight our ATMP development and production team with Annette, Naomi, Ivana, Ganesh, Ines, Flo and Scholt, and especially in the UMC Utrecht DRNL team with Simone Punt and Suzette Lauwe. And in all my projects, I'm working together with the UMC Utrecht cell therapy facility that's headed by Colin de Haar.
Thank you, Trudy. Thank you. I would like to thank you for this very interesting lecture. It's amazing how much disciplines and also resources you have to pull together, and you managed to pull together to create the impact of this novel technology. And I have just a question regarding to where we're standing at this moment, right? Before you gave the lecture, we were chatting, you mentioned that the breakthroughs happened around 2010. We are now 13, 14 years further down the line, and we see impact in a couple of cancers. There are also a couple of cancers where it still doesn't work, right? So that tells me we still don't fully understand how the tumor immunology works. Can you sketch where are the knowledge gaps and also where you think the field is heading towards?
Thank you for your question. Indeed, we are successful in hematologic malignancies, indeed, especially B-cell malignancies can be targeted very well, but also there we see that not each patient is responding. So we are really, I think the field needs to understand why a certain patient does respond and does not. And now, because these medicines are on the market, there's a lot of data coming available, which points in certain directions already. So that field there, that direction we are really going forward. But in other tumors, like you said, like solid tumors, we don't know yet which are the best targets for our therapy. So which antigens are the best targets. And there I think there's a big gap, like we try to fill also with our tags, which is the potential to work across tumor identities. But there we just started. So we just only treated a handful of patients, and we need to get much more data to be able to understand fully the complexity.
So we just started. You just started. So we can expect much more.
Yeah,
Probably you have to educate also new and novel people in this field.
Yeah.
Or it will be a thriving field. And I'm sure you will be in the center of that. Thank you very much for your contribution. And we're very honored to have you here at the UMC Utrecht.
Thank you.